Inside of our cells, our chromatin, our DNA, is in a particular structure. We've hinted at this before. If you remember, I said it's going to come up in this exam, it's going to come up, it was in the last exam, it's going to come up in the final. It's a common question. DNA is actually wound around a group of proteins called histones. Remember I made the joke, I said, what about herstones? They're called histones. We wrap the DNA around those. That's what stops it from getting tangled. Depending on how tightly DNA is wound, how tightly it's wrapped, that determines whether we make that protein or don't. If something is so twisted and so coiled that we cannot get our, I erased my picture. If we cannot get our DNA polymerase in there, or we can't get our mRNA polymerase, I'm sorry, let me rephrase. If something is so tightly twisted and coiled that RNA polymerase can't read the nucleotides to make the RNA transcript, how are we going to send it out to the protein? And so sometimes turning a gene off just means it's tightly twisted and coiled. And so when DNA is wound around this protein, wound up, we call it a nucleosome. That's a word that means both DNA and those histone proteins. So we have a bunch of nucleosomes. And we have a picture of it. Isn't that so cool? I think so. Look at that. That's wild. 10,000 times magnification. I'm just appreciating it for a second while you write your notes. If those nucleosomes, if the chromatin is kind of uncoiled, it's loose, we can see all the nucleotides, it's called euchromatin. EU means true. We call this transcriptionally active. We can use it to make a protein, and this is almost guaranteed to pop up on your exam. I know each of you have different exam questions from a pool of questions, but odds are it's very high that you're going to see this on the exam. Heterochromatin, hetero means different, is tightly packed, cannot be expressed, transcriptionally inactive. So euchromatin, we can make the protein. Heterochromatin, we don't. All right, in my last couple of minutes, let's learn something really, really cool. It's my favorite topic of gene expression. It's wild. If you have two X chromosomes, one is tightly coiled, one is euchromatin, one is not. It's heterochromatin. This tightly coiled X chromosome is called a Barr body, B-A-R-R, named after the person who found it. It's inactive. So imagine if you have two X chromosomes, if you could think, go magic school bus yourself into the inside of your cell, one of your X chromosomes is active, one is coiled. In humans, doesn't do much. It depends on what cell we're talking about. But a way that we can actually see this process is in calico cats. You ever seen a calico cat before, or a tortoiseshell cat? So they have some brown fur or some black fur or some gray fur. We think, why? How does that happen? And for the most part, it only happens in female cats. That is because the alleles that code for orange and the alleles that code for black fur are on the X chromosome. And depending on what cell we're talking about, one X chromosome is a Barr body and one is not. If your black chromosome is on the Barr body and it's coiled up, it's not going to be expressed. And so if your other X chromosome is orange, it's going to be expressed. So depending on what cell we're talking about, depending on which one's the Barr body, that's the fur color we're going to see. So it won't be a blend, it's going to be one or the other. Now fur genetics, if you ever take genetics, it's a whole chapter. Fur genetics is wild. The color isn't just based on one or two alleles. There are a ton of other factors. that determine whether it's dilute, expressed, white, clear, red. There are a bunch of stuff going on. But with black and orange calico, they're on the Barr body. And because this is my favorite topic, it's probably going to be on the exam also. Barr body is coiled up, and you see it and you don't. You can impress your friends. So here's my question, and I want you to answer it out loud. We have a couple more minutes here left. I'm not going to use this for attendance or anything, but I want you to answer back if you can. Critical think. Think back to the last unit that we just took, the last exam. And I'll open up chat in case you have some thoughts also. When could we see a male calico cat? What would cause that? There are a couple reasons. Think back to the end of chapter 10, when we were talking about XX and XY. Yeah.
It takes on male and it has the XY. Wait, female's XY, right?
Female's XX.
XX. So
And then male, for the most part, is XY.
but if it was a male and it had XX.
When could that happen? You're right. With the SRY gene issue. So there are two things that could happen here. There could be an issue like too many copies of the X chromosome. It could be XXY, Klinefelters or something. Or it could be an issue with the SRY gene. Cats are mammals too. And so just like us, they have the same regulatory factors that make external or internal genitalia. And so those that male cats could still be born XX and just have an SRY gene mutation. There could be an SRY on the X that could have migrated over. Isn't that kind of wild? And so it helps us see this process. We get stuck in our mindset that it's only XXXY. That's not really the way the world works. It's a lot more confusing than that. Most of biology is. There's a thing that I learned in SEMO, and it stuck with me forever. One of my professors said it. Life doesn't read textbooks. We're trying to understand it. We write the textbooks, right? But life doesn't care. Life does what it wants, and we just try to figure it out. But that's wild. People pay thousands of dollars for male calico cats, as an aside. Because it's one of those things where it's kind of a novelty, because most of them are female, and so people will pay to have. Anyway, that's my last tidbit of the day.